The plasma membrane is an important concept. It is called selectively permeable because it controls everything that goes into or out of the cell. It does it by its structure. It has very, very specific structure. The plasma membrane is called a phospholipid bilayer. Outside the plasma membrane is a superficial membrane of carbohydrates. These are fairly complex molecules. Look at the words. Proteoglycan means a protein with some carb. Glycoprotein, protein with carbs. Glycolipid, lipid with carbs. So we've got different kinds of molecules out there, and each has different function. Some of the functions would be cell recognition, how one of our cells recognizes that another cell is from our same body. Some of these functions would include binding to other structures, and some of them are for lubrication. This whole carbohydrate-based membrane kind of outside the plasma membrane is called the glycocalyx. So the plasma membrane itself, again, it's a phospholipid bilayer. It's selectively permeable. The brown areas are the regions of the phospholipid bilayer. The hydrophilic head is on the outside and water-loving because the extracellular fluid and the intracellular fluid are both water-based. So these water-loving heads are exposed to the water, and the fatty tails, each phospholipid has two fatty acids to form these tails. These are sandwiched together in the membrane to keep the fats away from the water. Some of the proteins that are embedded in this, all these structures here are proteins. Some of them are part of the membrane. This integral protein is part of the membrane. If you took it out, you would just damage the membrane. Some of them span the membrane. Some of them are just on one side of the membrane. So there are quite a variety of different kinds of proteins. Again, you can see some of these odd structures here are proteins with other components. So the glycocalyx had some lipids, some carbohydrates, kind of on the outer surface of that plasma membrane. Some of these proteins, look at that term, transmembrane. It spans the membrane, and in this case it's open. So some of these are open all the time. Some of them are closed until the right molecule comes by. So you should be able to draw the phospholipid bilayer with the hydrophilic heads at the outside and the inside, hydrophobic tails on a sandwich between the two hydrophilic heads. Inside the membrane are molecules of cholesterol. We make cholesterol, but we also eat cholesterol, so we can have high cholesterol if we eat too much cholesterol. But the cholesterol goes into our membranes to stabilize the membrane because the phospholipid bilayer is a bit like a soap bubble. Really, really flexible, delicate, and so it's stabilized somewhat with this cholesterol molecule. So when you eat meat, you're eating that animal's membranes, which also contain cholesterol to stabilize their membranes. So the plasma membrane separates the inside of the cell from the outside. It regulates exchange with the outside of the cell. It's sensitive to the environment, so it makes decisions somewhat on what's going to come in and come out of the cell. And it's really isolated by that lipid, that phospholipid bilayer. Very little will cross that membrane without assistance. Here are some functional classes of the membrane proteins. Anchoring proteins are going to anchor the cell to other structures. Recognition proteins determine whether the cell is a member of the body or not. And receptor proteins will bind to very specific molecules. Our cells have receptor proteins for cholesterol, for example. And the molecule that it binds to is called the ligand. Carrier proteins will help transport substances across the membrane. And channel proteins are channels that are open that will permit substances to cross. Maybe the substance comes into the cell, maybe a different substance goes out of the cell, but channel proteins would be required for this. In the membrane or skin of a cell, what makes the hydrophobic interior region? Those fatty acid tails are sandwiched between the two hydrophilic heads. To what part of the plasma membrane do the bones of the skeleton attach? Anchoring proteins. The cytoskeleton is an internal skeleton for the cell. We have an internal skeleton of bones. The cell has an internal skeleton made of proteins. Here's a diagram, and right next to it is a electron micrograph of those proteins. So there's several different kinds of proteins. There's microfilaments, which are made up of actin. You may recall the microvilli, or the cellular extensions, to increase surface area. These have microfilaments traveling up through them as a core. We also, in the cell, we also have what's called a terminal web, and it is a layer inside the plasma membrane of these proteins, stabilizing the cell. Intermediate filaments are very strong, and microtubules are the largest. The interesting thing about microtubules is they can form and unform as needed. It's almost like saying, well, if we needed an extra bone to walk a different way, we would just grow it and use it, and then it would go away. This is what happens with microtubules. They form and unform as needed. Centrioles are that cylindrical structure necessary for cell division. They're made up of microtubules. There are 2 of them in each centrosome. And without these centrioles, the cell cannot divide. Plants can divide without centrioles, but animal cells cannot. And there are the 2 centrioles at right angles to each other. And that structure, the 2 of them together, is called the centrosome. Before the cell divides, that structure will divide. Cilia are very long extensions. They will beat rhythmically on the exterior surface of the cell to move fluids or secretions across the cell. We find cilia in our respiratory tract, and the purpose of that is to move substances. As we sleep, we inhale substances that must be brought up our throat, and then we swallow it back down into our digestive tract. We cannot allow any particles to get into the lungs. And you can see that the cilia are composed of microtubules. And this is just showing the action of a beating cilium. Cilium is singular. Which of the cytoskeletal components has the largest diameter? Microtubules are the biggest. What cytoskeleton component is involved in cell division? The centriole. Ribosomes are the protein factories. They are essential for protein synthesis. They're produced inside the nucleolus, which is inside the nucleus. It's composed of 2 subunits, and each is made of ribosomal RNA and proteins. Some of these ribosomes are attached to the ER. This makes it rough ER, and some are free throughout the cytoplasm. The ER is a network of tubules. And there's 2 types of ER, the smooth ER, which is for the manufacture of carbs and lipids, and the rough ER, which has ribosomes attached to it, and that's why it's called rough ER. That is for the modification of proteins for export. Smooth ER A little more detail on the functions of the smoothie are synthesis of phospholipids, which are found in our cell membrane. Synthesis of cholesterol, which is also found in the cell membrane. Synthesis of steroid hormones. Steroid hormones use cholesterol as their base. Steroid hormones are hormones like estrogen and testosterone. SER can produce and store glycerides in the liver and fat cells, and produce and store glycogen in skeletal muscle and liver cells. Glycogen is our storage form of glucose. It's long chain, it's just many glucose molecules. This is showing what happens to a protein as it's being produced. It's modified, it enters the rough ER, and it's packaged for export to the Golgi apparatus. Again, the functions of the rough ER include synthesis of proteins. Now, a non-completed protein, a protein that has lots of amino acids but doesn't have its final shape, is called a polypeptide. So when that protein is completed, there are many things it can become, and examples are enzymes or glycoproteins. If they are for export, they will be transported to the Golgi apparatus in transport vesicles. Here's our Golgi apparatus, a little bit like flattened pancakes, and the vesicles carrying the modified proteins from the rough ER come to the Golgi apparatus, fuse, and release the proteins inside the Golgi apparatus for more modification, for packaging, for concentration, and then it will secrete vesicles with these completed proteins for export from the cell. Don't worry about the cis and trans phase. So products from the rough ER arrive in vesicles, they fuse, they enter, they're modified by the Golgi apparatus, and they leave, heading over to the plasma membrane for export. So some other things that the Golgi apparatus can produce are, it can produce more phospholipid membrane to increase the size of the plasma membrane, to add to it, to repair damage to the membrane. It produces secretory vesicles for secretions from the cell, and it produces enzymes such as those in the lysosome that are used to degrade bacteria or old organelles. The lysosomes themselves are kind of interesting. Filled with about 40 different enzymes. They may be used to destroy the cell itself. If the cell is damaged, if it cannot be repaired, the lysosomes will open up and destroy the cell. They destroy old organelles. And when we developed as a fetus, we had webbing between our fingers that lysosomes are programmed to break down. So you can see the fingers of the fetus, how they are at first webbed, and then eventually that webbing will break down. Sometimes the webbing doesn't break down. Often, as if children are born with that webbing is cut. Here you can see kind of a cute tattoo where the webbing has not been cut. Tadpoles, their tails don't fall off as they develop into frogs and toads. Their tails are reabsorbed because lysosomes are programmed to destroy that tail and reabsorb and reuse those nutrients. And here are some functions of the lysosomes.